next speaker is Andrea Luppi from University of Oxford in UK.
All right, thank you for being here. Let's get started. So as neuroscientists, we are all interested in the question of this organ. It's the same organ, it's the brain, but many of us will look at it at different scales, different levels of organization. And one of the things that I'm really interested in is how we can combine them instead. And one way to do that is through pharmacology. And in particular, I like to use anesthesia because it is a small molecule that acts locally but has global effects on brain and behavior, and it is very conserved across species, from humans to nematodes, mouse, macaque. So we can use it to combine these different scales and bridge them to understand how molecules affect the overall functioning of the brain. But here comes a problem with the usual way that we approach this, because yes, we can put people in the scanner and compare the brains while they are anesthetized and while they are awake and try to find differences there. Many ways of doing that, but the usual story is missing one key point, that those two time series that we might be observing from the same region are different in all sorts of different ways. Is it about their amplitude that we should care about? Should we instead care about their autocorrelation? There's a lot of different features of time series that we can handpick, whether it's autocorrelation, the distribution of the time series, and so on and so forth. And so rather than handpicking features, we thought, why not just throw at these time series everything that anybody has ever thrown at a time series across the neuroscientific and the scientific literature more broadly. So we're not talking about 1 or 2 or 10 features, we're talking about more than 7,000. And this is a method developed here in Australia by Ben Fulcher called highly comparative time series analysis. Take everything and throw it at the data and see what sticks. It's as simple as that. And then the question becomes, how do we make sense of this? So here's the approach that we take. We scan people while awake and while anesthetized, same as the regular story so far. And then for every region in their brains, we take that time series of bold signal that we've obtained, and we extract 7,000 features out of that. We do the same when they're anesthetized, and we compare those features. We ask which features change in the same direction, either positively or negatively, under anesthesia. And I've said that one of the great things about anesthesia is that it's very conserved across species. So we don't do this just in humans, but we also do this in macaques, marmosets, and mice, using different anesthetics, because anesthesia, one of the other things that's interesting about it, is there's not just one drug. It's a lot of different drugs acting on different molecular mechanisms that nevertheless eventually end up at the same endpoint. And I did mention C. elegans, and in fact, we don't just use functional MRI in these species, we also use single neuron calcium imaging in the nematode, again, anesthetized. So we combine the data-driven approach of throw all time series features at our data and see what sticks, with the hypothesis-driven approach of we expect that there should be consistency among some of these features because we know that ultimately all of these anesthetics have the same endpoint in these animals. They lose responsiveness, they become behaviorally unresponsive. So is there any consistency? Well, there is, because we can see the features here are all ordered in the same way. And as you can see very clearly, some features consistently and systematically drop their values when animals and humans are anesthetized, and then other features increase their value when animals and humans are anesthetized. So what kind of features are there? In fact, we do find that there are about 500 out of those 7,000 features that are consistent. This is significantly more than you would expect by chance. And overall, we find 2 big clusters of features. Some of those pertain to the predictability of the signal. The signal becomes less predictable in terms of its future by looking at its past. And conversely, we see that the signal is overall less autocorrelated. In other words, the brain seems to be losing its memory of what was happening just before as you're undergoing anesthesia. And this happens across a whole suite of different measures of autocorrelation, linear and nonlinear. And here is an example of what is happening to those time series. You see the beautiful long autocorrelation all of a sudden drops much, much faster under anesthesia. This is the same across all of those species because all the features that you're seeing here are changing consistently in the same direction across every species and across every anesthetic. But I did mention that one of the other things that I'm interested in is bridging all of these scales. And we know that ultimately these local effects that we are seeing in terms of changing time series features are being acted upon by the drug by acting on neurons and their receptors, acting on molecular machinery that is ultimately coming from genetics. So we can look at gene expression that fortunately we have for the human and for the mouse, but also recently for the macaque. Gene expression across, in this case, about 80 brain-related genes that we have in each of the 3 species, and we can ask, does a particular pattern of gene expression explain across each of these 3 species and across each contrast the relationship between changes in local features and changes in gene expression across the cortical mantle? And we find, even after controlling for the role of spatial autocorrelation, which, as we just heard, can become an issue, we find that there is this beautiful anterior-posterior pattern that is shared across humans, macaques, and mice, and that loads highly onto genes pertaining to the control of sleep-wake cycles, such as orexin, and on the pValv gene, which is the main marker of parvalbumin-positive inhibitory interneurons, which happen to be one of the main targets of the GABAergic anesthetics like propofol, sevoflurane, and isoflurane, all of which we included in our data. Now, we also know, since we have been looking at the loss of autocorrelation, we also know that autocorrelation and, in general, the temporal intrinsic timescale of the brain is not uniform across the cortex, but rather across many different species, including humans and macaques, we know that there is a temporal hierarchy of intrinsic timescales. And so we were wondering whether that can explain the changes in temporal autocorrelation that we see in our data. Are they uniform, or are they differently distributed across the cortex? And we find that, in fact, across all of these different species, the intrinsic timescale of the region or neuron at rest during wakefulness is predictive of how much the loss of autocorrelation will be. And in particular, you can think of this a bit like progressive taxation in the brain. The more you have, the larger your autocorrelation at the beginning, the more that will drop under anesthesia, regardless of the anesthetic, such that overall, the hierarchy of temporal intrinsic timescales is becoming compressed under anesthesia. But in addition to knowing that there are local changes, which we have now systematically mapped in the brain, we can also ask what are the relationships between different regions, because we know that those are also changing. And so we can combine these 2 approaches. We can ask what kind of manipulation using a whole brain computational model will produce, despite being a global manipulation, will produce the same kind of local patterns that we observed systematically across anaesthetised species. And we find that the best way to do this is to weaken the effective coupling between regions. In other words, you weaken the ability of one region to causally influence another across the entire brain, and as a result of this global perturbation, you see these local changes that we observed under anesthesia across all of these different species, as well as an overall drop in the synchrony between regions, which we also observed in each of the species under consideration. And at this point, we've looked at one way of trying to identify a causal mechanism with computational modeling, but there's also another way, because we can say, what happens to all of these signatures when we reawaken the animal using thalamic stimulation, despite the animal still being pumped full of anesthetic? And this is work that my colleagues in Paris carried out. And what they find is that depending on which thalamic nucleus you stimulate, the monkey either remains completely anesthetized or he wakes up. And in the case where you stimulate the correct nucleus, the central thalamic nucleus, you reawaken the monkey, and all of those signatures that I've been describing, they all go away. So the monkey's brain, from the perspective of temporal dynamics, looks awake, and the monkey looks awake and behaves like it's awake, even though it still has propofol in its bloodstream. So these signatures do not track the presence of the drug. They track whether the animal is conscious. They do not track the presence of electricity being inputted into the brain, because we have a control condition where we stimulate a different thalamic nucleus and nothing happens. They track whether the animal is conscious. So overall, there are two main patterns that we can discern from this purely data-driven analysis of the large-scale dynamics of the brain across from single neurons to whole brain. First, regions become temporarily isolated from their own future. The memory of what's been happening is lost, and the autocorrelation shrinks. Second, regions become less able to talk to each other under anesthesia. There is a desynchronization overall. So spatiotemporal isolation is the name of this game. And all of this is under potential local thalamic control. So altogether, I hope I've convinced you that we can use dynamics as a kind of Rosetta Stone to bridge from single neurons in the worm to whole brain in the human, and everything in between, and from biological brains to artificial brains, such as in our computational models. Now, of course, I cannot claim to have done all of this work in isolation, far from it. In fact, it takes a whole village, a synergistic effort to make this happen. So my thanks to all of those who have contributed to it. And you can read the full story in this preprint, scan the QR code to get to it, or if you want, talk to me by email or social media. Thank you all for your attention.
Okay, we have some questions in the response box. So the first question is, you reduce the spatial complexity by using an atlas, but increase the spatial complexity. What if it is the other way around?
I'm assuming the second part is, but increase the temporal complexity?
Yes, I mean the temporal.
Right, yes. I'm not sure how one would do that. Oh, well, actually, yes. So you could expand by looking at voxel-wise, for example, I suppose. People have done that a lot. There are a lot of papers out there that look at, voxel-wise. I've done some of them. Voxel-wise analysis, looking at one or a few different temporal measures. My papers and those of many others have been carried out. This approach is based on the fact that using the temporal dynamics, it's the Rosetta Stone that I'm interested in. Voxel-wise analyses are different in every species, but those dynamics are the same. So you can use them as a common space and find what's similar. That's the point.
I just have a sort of a follow-up question on this. So is there some patterns you saw that across aging or developmental trajectories in the species, in individual species, and is it conserved across species?
Yes, so we did not look at different ages in this data, but there's a beautiful paper just came out in PNAS, I think, a few months ago, in 2025, looking at MEG data across the aging spectrum, and they were also finding, using HTSA, and they were also finding changes in the autocorrelation in particular.
Oh, okay, thank you. So in the interest of time, I will request the other questions to be directed to you after the talk, but I will request the next speaker, Francis, so for the last talk. Thank you.
Maybe you see the one who's from.
Yeah.